because I hope to get to the animals today. Okay, so we had talked about the chytrids. Now, let me ask you a question. I was thinking about this before class started. The chytrids are really small, quite obviously. That little bar there says 25 microns, so these things are microscopic. So let's imagine that you were a biologist or a researcher of some type, and you notice these organisms underneath the microscope, and you saw these little stringy things that were hanging off. Is my pen on? I decided for a different color today. I'm going to go with red. How would you know these things that are, oh gosh, how would you know these little stringy things there are hyphae and not flagella? What would you look for? Critical analysis of information. How would you know, or what would be your cue? Cue, clue, whatever, same thing. Typing. Anybody else though? Come on, there's something we said that's very distinctive about fungi. We said it's very distinctive, in structure. So think about that. Oh, goodness. Spencer, you know what I'm talking about. Nicole, right? Yeah, everybody's got cytoplasm though, and yeah, the hyphae could be continuous or could be septate. Yeah, but I don't know if that would really tell you that was a flood. I don't know if that would distinguish it. There's one thing that one of them has, or there's some structure or some material that the other one does not. And I'm asking you to go back to Bio 1, I know, because you talked about flagellal structure back then, but think more maybe in what I've told you that's very interesting about how fungi are constructed, how their cells are constructed, how their structures are constructed. Get the wrong pen. Gotta be careful not to write on the screen with that. Oh, okay, maybe, but I don't know which way is posterior or anterior in that thing, do you, on that chytrid? So if I'm assuming that there is a posterior or an anterior and a posterior in that thing, I'm not sure I would know which one is what. Would you? I don't see any eyes or anything like that, so it'd be hard to tell. I guess you could watch the motion, but I don't know about that either. Yeah, it is a good question, right? I should put it on the exam. All right, all right, I'll give you a hint. Animals, certain animals, not all animals, but certain animals have a structure they share with fungi that ties fungi to animals more closely than it ties animals to plants. What is it some of us animals have that fungi do too, that let us know that somewhere back in that branch that it worked kind of like the plants split off and then the animals and the fungi split off? There's a number of characteristics, but everybody's got not everybody, but not mitochondria. Plants got mitochondria, animals got, okay, let's go back to the original question again, right? Look at the hyphae, and I'm saying there's something that hyphae contain that you may or may not find in flagella. The hyphae are those long, continuous filaments of fungal cells, quote unquote, right? They can be septate or they can be synocytic. They can be open. But what is the cell wall made up of in a fungus? What is the structure? What is the material? Yeah, there you go. Chitin. Who else has got chitin? No. Bugs, reptiles. I was reading yours. No, arthropods. Okay, don't just say bugs, because crustaceans, which aren't bugs, which aren't true bugs, which aren't insects, okay, also have chitin. That's what their exoskeleton is made of. Okay, now, do flagella, going back to Bio 1, do flagella have chitin? Are flagella made of chitin? No, 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 no. So if you looked at that under a microscope and you said, I don't know, wait, is that hyphae or that those flagella? If you could figure out whether there's chitin in those structures, then you would say, oh, this is a fungus. This is not a miniature animal or like a paramecium. Because remember, just because something's really small doesn't mean it's a protozoan. Like for example, rotifers, which we'll talk about in the animals. You need a microscope to see them, but they're true animals. Okay, so all right, see that detective work? How we do that? Right now, of course, there's millions of other things that are going to either support or even refute these hypotheses. Remember how hypotheses and theories always work, but that's why we point to those when we go look at those. Those aren't flagella; those are hyphae. Okay, so let's jump on down. We did the glomeromycetes, didn't we? Yeah, we did the glomeromycetes because remember those are really important for mycorrhizae, which you saw under the scope the other day. Mycorrhizae, here we go. Right, mycorrhizae, don't I have a picture of mycorrhizae? Or did I just show you one in lab? I might have. Remember, mycorrhizae were the, I guess I better change color, where when you looked at it in the microscope, you looked at a root, and you were looking through a cross-section, like here's the root, right? And they took a special thing, a knife, a sectioner, and they cut a little thin section. And then they planted that down, so like a hamburger patty. So that's what we mean by an excess or a cross section, or maybe that means extra small, but whatever. In the microscope the other day, when you look, you saw the xylem and the phloem, you saw the conductive cells, the vascular tissue, blah, blah, blah. But there was, and nice about staining in microscopes, right? Because on the outside, everybody said, where's the mycorrhizae? Remember I said, look on the outside, it's that blue layer. And if you looked closely enough with your 40x objective, you might have even saw that the blue layer was actually fibrous, maybe. Okay, so that's the mycorrhizae. So that's what we mean by that. So mycorrhizae, of course, like we said, are very important for plants because they're the ones that actually create that real rich nutrient soup that plants can grow in. And that it really was beneficial for moving out on land, wasn't it? So you notice so many times in organisms that many times the evolution of organisms works in conjunction with one or more participants. It's what we call coevolution. Don't confuse that with convergent evolution. Everybody knows what that means. Of course, we talked about it a million times. But coevolution is, well, in a good example, predator-prey relationships are coevolution. What do you think would happen if a predator become extremely good at hunting its prey? What do you think would be the potential consequences of that? And there might be more than one too. So. Yeah, that's right. And then what would happen to the predator, especially if the prey that's being hunted made up a big portion of its diet? What would happen to the predator? Yep, and that does happen too. It's really kind of cool. Well, it's not so cool for those that are dying, obviously, but ecologists, folks that look at things like relationships, predator prey, you can plot it. So I'm gonna draw. So like there's a peak in the number of prey, okay, and then soon after that, because there's a lot of prey, a lot of reproduction is going on in the predators, and they peak, and then as they peak, the prey go down because guess what? There's a lot of predators. And now as the prey go down, there's less to eat. So it's an out of phase sine wave, if you remember what a sine wave, if you know from math, right? So it's kind of they're both in the same shape, but they're moving sort of out of phase with one another. It's really interesting to watch. So that's what we mean, but kind of by coevolution and ASCO, or I'm sorry, Arpuscular mycorrhizae and the other type. You looked at ectopic mycorrhizae, or ectopic. No, I'm sorry, ectomycorrhizae, excuse me. Ecto means on the outside, right? See, remember, arpuscular mycorrhizae form these arpuscules, these little, what did I say it looked like? I don't know, splattering or something like that, right? Okay, let's go on to new stuff. Any questions about that? I got a lot of new stuff to talk about, really cool new stuff. I love fungus, you know, just because they're so weird. I mean, animals, it's always animals. Anyway, all right, cup fungi or sac fungi. They're called cup fungi or sac fungi because because they make a cup or a sac. Even the little thing, this thing over here, the morchula. Morchula is a morel, M-O-R-E-L, morels. Why did I just write it? It's right there. There we go. So morel. So ascomycetes or members of this group, the cup fungi, and asco is cup again. Remember angiosperm meant container of seed. Ascomycota, mycota is fungus, and asco in this context is a cup. So the morels, even though it doesn't really look like the one below it, which I didn't put the name, those are also cup fungi because look, it's made up of a bunch of little tiny cups, isn't it? So this one's one big cup, but these are little tiny cups. Same thing then, right? And this one right here, the Tubermelanosporum, those are those the truffles? Can't remember. I pull these pictures offline and I'm trying to remember if those are the truffles. I think they are because I have a picture of them on the right. Yeah, those are the things that they use the pigs. Does anybody know what there we go. Let me look up that species again. Tubermelanosporum. Are those the truffles? Oh, what is there somebody's name by that? What does that mean? Yeah, truffles. Those are those delicacy, right? Those things that people look at the price over here in the lower right. I don't know how well that's showing up, but that's for what, 8 ounces? Is that right? $580. Holy cow. Weren't we talking about like champagne the other day? And I said, really, do you think $1,000 champagne is worth $1,000? Would you pay $600, almost $600 for 8 ounces of mushrooms? The one on the right is another sac fungus. I don't think that's yeah. Holy cow. Has anybody ever eaten them? I don't think that's not a truffle. Somebody in one of my classes had worked at a restaurant said they use truffle oil. Like you can flavor things with the oil, but I'm like, I don't know. I can't imagine it tasting so good that I would pay that kind of money for it. You know why they use female pigs, right? To find them. The female pigs sniff them out because they grow in leaf litter, so you can't see them. So the female pigs sniff them out, and right before the female pigs begin to consume them, along comes the dude who's collecting them. Says, "Oh no, thank you very much." But it's because of this thing called pheromones. Pheromones are like I always say they're kind of like external hormones. What I mean by that is they're chemicals that are released into the environment that relay signals. And one of the things they might release is sexual receptivity. Like a male and a female trying to attract one another might release a chemical to the environment. You know, some folks would argue that humans might have that. Maybe you call it, oh, it was just chemistry. Like, I don't have the record. Yeah, I do. I looked at the wrong button there. It sometimes even like online you can find it pheromones, and it's just, you know, you can put it on and males just can't stay away from you, or maybe it's the other way around or something like that. Yeah, maybe. I'm not sure humans really can smell that well to be able to do that, but female pigs can sniff out male pigs, and so that's what the truffles smell like to them. So and then they go, oh, some neat. So that's what the dude behind with the leash on the pig says, no, that's a $300 or $500 fungus. Holy cow. The stuff that grows on trees that we've talked about several times, lichens. Remember, a lichen is a great example of a symbiotic, like what we would even call now that you guys are, I introduced these terms to you. Maybe you know already anyway, but obligate, right? Obligate symbiotic relationship, right? They got to have each other. You tear them apart. They've lived together for so long. They've actually got their own name. They're even got species, Okay, there are, you guys, if you're identifying your lichens, you find out it has a genus and a species. And if you think about it, you step back for a second, boy, that's really messes the whole thing of species up, doesn't it? From what we talked about early on, right? Because these are two different critters. A green algae, which is a plant, kind of a protozoan, and sometimes even a blue-green algae, which is a doggone prokaryote, a cyanobacteria, will live with a fungus. They live in this relationship, and that's what we call a lichen. About a quarter of the fungi that do this are these sac fungi. And there's some pictures of lichens on a tree. I either got those up online or I took that because that's got to be ball moss. That looks like what was that, Christmas tree lichen? There's one that's red and there's one that's red with the green around it, or Christmas wreath lichen. Okay, lichens are everywhere. Okay, now let's look at the reproductive part of this. Now remember the really significantly different thing about fungi in general is this purple part of the cycle that you see over here, right? Remember that I said that even though all cells technically do this plasmogamy and karyogamy, what fungi do is they extend that time in between. So they have this, heterokaryotic, or in the of these kind of fungi, where the hyphae are septate, what you might end up with is dikaryotic hyphae. Okay, since they're septate, you do have that division between them, so you can kind of get the idea of a cell. But remember, in fungi, it really is still all about the nuclei. So that's why you got this sort of purple range. So you get two different nuclei. Now, they don't fuse. The cells fuse, plasmogamy. So you get this thing called an ascus. Okay, the name, we're trying to, or what we're doing is keeping the similarity of term. Ascus forms an ascocarp. The ascocarp is the thing, the actual moral or truffle itself. That's the ascocarp. The little in the moral, it's the little pockets that are in the moral. But those things form an ascus. Now those are dikaryotic, you see. So I'm jumping back and forth across the image. I'm sorry. So we're on the right now. So dikaryotic would be different than heterokaryotic, and you'd probably be heterokaryotic because of the fact that your hyphae are synocytic. They're open. There's no septate between them. Now let me jump back for just a second, like I do with plasmogamy, and show you how that comes about. Let's get this easy part out of the way. There is also asexual reproduction. Okay, and asexual reproduction, I say the easy part is because it basically is spores that germinate in the hyphae that make more spores that germinate in the hyphae, so on and so forth. I do want to point out one thing, though. Spores can be made two different ways. Okay, down here, those are spores. If they're spores that are produced asexually, we call them conidia. Conidia spores. Conidia spores are asexual spores. We differentiate that from regular spores that are made down below, that I got the arrow pointing to. So the conidiophore is the thing, like the sporangiophore in black bread molds. Phore is the structure that makes these conidia. Like the sporangiophore is the structure, that stalk that has the sporangium. The conidiophore is the structure. There's really not a conidiogym, okay? But the conidiophore is the structure that produces the asexual spores. Just remember, conidia, asexual spores. If it doesn't say conidia, then it's sexual produced spores, the typical spores. All right, two of them come together. Now what really happens is one from one mating type kind of sticks to the end of the hyphae of another. See what that is right there? So it's kind of like the pollen, remember, sticking to the stigma? And guess how they do that too, by the way? Pheromones. Pheromones. Those mating types release pheromones. The little red thing sticks to the blue thing, right? And they fuse together in plasmogamy, form a dikaryotic. Now remember, staying in that purple stage for an extended period of time, unique to the fungi, is good because you get the benefits of being diploid. Probably with less interaction of the dominant and recessive going on. So you get the benefit of more genetic information. You also get the chance for more genetic recombination. Now, genetic recombination is a term that we use for many different things. You can do it in the laboratory. Genetic recombination is what viruses could do to bacteria. Remember how they can inject genetic information? Anytime you mix up or insert or move around genetic information, you're doing genetic recombination. And one of the things that the chromosomes can do in genetic recombination is they can cross over. Remember in meiosis? They cross over different places. So they can keep doing that. So you can get all different kinds of ASCII, plural of ascus, many different kinds of ASCII. And then eventually the nuclei do fuse and you get that zygote. Now here's another big way they differ than animals. You're the result of a zygote, okay? You're a grown-up zygote. You're a multicellular representation of that very first cell, the diploid nucleus. Mitosis, 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 things fall apart, leg falls off, well, you don't really grow in the leg with mitosis, but a wound, you get mitosis, right, okay, now. But unlike animals, fungi use that for one reason only. They use the diploid stage. They abuse, well, they don't abuse it, but they use the diploid stage and they say, I just need you for a second, okay? I just need you to do something. And what I need you to do, diploid stage, is just exist around long enough to do this. Thank you, Penn. Long enough to do that. Why? Why? Someone tell me why. Why is what I'm saying so significant? The zygote is made just to undergo meiosis. Why is that so doggone important? There you go. Genetic variability. And remember, when they're in that dikaryotic hyphae, they're all mixing up. So my goodness, you're rolling the dice. Now, when you looked in lab the other day, you may not have seen it because, when they die, the spores, and in real life, nothing ever looks like it. Like someone I think in lab said, why didn't the penicillium that you're looking under a microscope and the aspergillus look like those pictures I had? And I'm like, yeah, well, because we don't have super expensive imaging devices. Wouldn't that be nice if it does? But if you do remember under the microscope when you looked at Peziza was that one that at 4x, it was like a cup like that, okay? You remember the little fingers when you looked at it higher magnification. Those were the ASCII. And if you looked inside, there were lumps. Some of them might have been dyed, and you might have seen maybe not, but each one had 8. And that's real hard because sometimes not all of them develop right. So theoretically or optimally, each one would have 8. And those are all ascospores. And ascospores are just spores, folks. They're spores produced by an ascocarp. So the ascocarp, again, is the thing. It's the mushroom. And those spores, those ascospores, differ from conidiaspores because now these are produced sexually. If some of you have a pen, draw on your screen, point to where the sexual part that I'm talking about happens. Where does the biological definition of sexual reproduction occur? Point to it in this cycle where it happens. Let's see how well I've emphasized that, guys, or emphasized to that, y'all. So nobody can draw on the screen, or nobody knows the answer. Because you can use your finger too, by the way, if your screen is a touch screen. Who drew that? As soon as you mix, now, if I was going to say technically, you're correct. Okay, you're correct. But I would say now technically go just a little bit further. The actual part that's occurring, the sexual part, does, biologically speaking, DNA-wise speaking, occurs right about there. But I think you probably meant the same thing. You meant when the two genetic informations come together. But the actual biological part of it occurs right there at karyogamy. That's why we call that sexual. Remember, asexual, that doesn't happen. You have no mixing of genetic information. So that's why these ascospores are sexually produced spores, right? So a little bit of terminology review there. One ascus, several ASCII make ascospores. Ascospores are made in an ascocarp. that all of these are found in the hole in the bottom of the sea. No, all of these are ascomycetes, sac. All right, now, let me ask you guys a question from lab the other day. Take a look at that ascocarp that's right there. What type of an ascocarp is that? What type of an ascocarp is that? Yeah, I asked you guys, there were two types that we looked at. There's actually three, but I only had you guys look at two. This one, by the way, is the opposite of Peziza. I'm going to do that. I just know that's Peziza, remember? You guys are flipping through your notes looking for the, or your lab notes looking for the name of it. Oh, well, I will tell you that this was the one for Peziza. You remember the one for the one you're seeing there? Oh, there's a way to remember it. Thing's clicking. What is that? Come on, nobody? Anybody know what that root word means? Para, like, oh, here's one for you. Where's the Earth during this? Where's the Earth during that? In relation to the sun, where's the Earth? Oh my goodness, 8th grade astronomy. No, yes, of course it's around, Spencer, but I'm talking about a relation to the sun. Further or closer? Yeah, closer, right? Perihelion means closer to the sun. So now imagine in this, using the same root, if I told you that the thesium is the opening. The opening of the sac. A sac has an opening. It might be a big opening or a little opening. But what if I told you thesium was the opening? Now, again, keeping that in mind and what peri means, what type is the one you see here? The ascocarp that you see here. Drumroll. Nicole, Jennifer, anybody? Marcelo? Thesium is opening. Peri is close. I've either lost connection or y'all just fell asleep on me. What is ticking over here? Why question? Why question mark? You told me what, didn't you tell me what perihelion? No, Mark did. If I told you that peri is close, thesium is opening, then it, yes, say with a, yes, that's a misspelling of paramecium, parathecium, thesium. And then, by the way, apposition in astronomy is the away from, the opposite side. So apo would be Peziza. So that would be the bigger opening. Okay, that help? Sometimes remembering those roots, because you remember a lot of these science words are all Latin based. That's why even though in high school I was like, I ain't taking Latin. I took French. I don't remember any of it though. But it's nice if you do have some idea what the roots mean, because they mean the same thing everywhere. I just can't think of one right now. It's not paramecium. No, that's fine. Peziza, apothecium, right? Remember when it was like 4x under the scope? It looked like a blue, it was kind of a blue melon-shaped object. And that's when you zoomed in, you could see that it was all these little fingers. Right? Okay, good. All righty, now let's go on to the club fungi. You looked at those two. Don't confuse, but it is very often the one you see. Club fungi, it's not because they often are toadstools, right? Toadstools, like that's the one where the cat, right, in Alice in Wonderland, very, 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 very odd story. Of course, a lot of those guys were all doing opium back then, so that's why they wrote that kind of stuff. He also had a really nasty habit. Anyway, the cat sitting there disappearing, the Cheshire cat. Okay, that's your typical toad. So it looks like a club. So that when I tell people your typical Basidiomycete, the club fungi is your toadstool, I'm like, that's one of the ways, but they're not all that way. Puffballs don't look like that. I got that one in class that I showed you. See all those spores? Holy cow. Shelf fungi, which doesn't look like a club at all. These really cool maiden veil. Look at that. Isn't that neat? So I see fungi aren't really regulated to any shape. Even the stuff that you see growing on the corn, those are things that we call smut. That's smut. Smut is a bad thing, okay? Obviously for the corn. The stuff on the plant, on the leaves, I don't know, it looks like an elm or something, maybe sycamore. That's called rust, right? These are all examples of club fungi. These are really important decomposers. They're very good at breaking down that lignin. Remember that organic substance that's really hard for living things to get? It's got a lot of carbon in it. Remember, fungi is one of the only ones that can break down lignin. We also have some prokaryotes that can do it. Mycorrhizae, they're important for that. Now, back to the smut thing for a second. I think I do talk about this later. The smut that you see growing on the corn, that's obviously not good for the corn, but that's actually a delicacy. Yeah, I do talk about it later. I'll talk about that again. It's actually a delicacy for us to eat. Where does now notice all fungi have asexual reproduction, but among the club fungi, they rarely have asexual reproduction. That's why it's not even here. And if it did, it would work just the way you can imagine, asexual, just fission. But it rarely do they reproduce that way. They most often reproduce sexually. That's why the cycle here. It contains both, but you could see this is the dominance. All right, let's start back up with that weird fungus thing again. Dikaryotic mycelia. So now it's a little different. The two mycelia grow together. It's not like a spore of one attaches to the end of the other, like we saw in the other one. Extended time, more genetic recombination. And what grows out of this is another carp. Carp is the fruiting body, okay? You have an ascocarp, and in this case, you have a basidiocarp. Now, that basidiocarp, like I said, that's the thing that we often think about. Oh, there's the mushroom. There's the fungus. But remember, most of it's underground. These things can grow really rapidly. That's the thing. That's the honey mushrooms. Remember, it's the mycelium that's 2.5 miles. You don't have this big toadstool sticking up. Boy, that'd be cool to see, though. I don't think you could structurally hold it up. When you looked at it under the microscope, this was Coprinus. Anyway, when you saw Coprinus in the big part of the microscope, come on, there we go. The big part, the scanning, it was round. Remember, it wasn't really like that. It was rosettes, the gills, but they were. You remember, it was round like that. I'm sorry. Again, they took the toadstool and they sectioned it just like that. So what you were looking at along each of those little rosette-looking things was what's in the blue in the picture. Remember, this picture looks familiar, doesn't it? It's what you saw under the scope. Okay, those are what you would find on the edges of the gills. Okay, that's why it's real hard to do it with those doggone mushrooms that you buy at Publix, because they've kept them cool for so long that the mushrooms aren't actually producing spores, so they very rarely can you see it. But the actual name club fungus, back to the original thing I said about that, they often look like toadstools, but they're not always, is the actual thing called a basidium. Basidium means club, so I could hit you with a basidium, I guess. All right, and the basidium in that picture you looked at was the projections, the little lumps in blue. See that? A little better in that picture. Some of the shots on the scopes were pretty good. Little lumps in blue. Now, what holds up those little lumps in blue, this little thin part right here, is called a sterigmata. I just think that's a cool word. At the very end of that, are spores. Not this time, not ascospores, still sexual though, but this time basidiospores. One of the questions in your lab said, where are the spores produced in sac fungi, the previous one we looked at, produced, versus where they are produced in club fungi? And the answer back to the sac fungi is inside. It's inside the asci. These are produced on the outside, see? They're produced on the outside of the basidium. This is the spore-producing structure. It makes the spores, and it's attached, yes, but it's on the outside. Okay, so again, a basidiocarp, the extended, and in this particular case, basidiomycetes, club fungi, they have really, really, really long dikaryotic stages. The purple part's really long in their case. A really short sexual stage, really short karyogamy, not sexual stage, I'm sorry, wrong word. A really short zygote stage, each ascocarp making a zygote. And then meiosis, so we're back to variability again, and we're making basidiospores, right? Sexually produced spores, sexually produced. They disperse, germinate, mating types find each other along the crossway of the soil. They release pheromones and they find each other. I wonder if there's certain pheromones they like more than others. Like, wow, that's a very sexy pheromone there, mating type. I'm not sure how that works. Weird critters, right? Now, as I said, these basidiocarps grow really fast, right? Because what happens is everything's underground, doing its thing, doing its exozyme thing, and then the conditions come right, like maybe it rains, okay? And the mycelium or the fungus says, wow, this is a great time to make spores. I'm going to get up and make some spores because it's rained and the conditions are really good. You know, or sometimes it's like, this is a good time to make spores. Looks like things are getting rough. You remember I said, you know, some species, or many species actually, can kind of, you know, they'll sexually reproduce, or they'll decide it's time for variability when things are good and when things are bad. Because when it's good, it's like, whoa, numbers, let's go, let's go, let's go. Things are good, things are good. Like, can you imagine those first plants when they crawled out on land, especially the ones with mycorrhizae? Holy cow, if they could have talked to, like, go, 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 you know, because the land's fighting back and, you know, all this sort of stuff. And thank goodness the insects came along. All right, so now what I mean is, okay, everything works real quickly. Now, these gills that they produce, back to the spores for just a second, make lots of spores, right, for reasons that I'm sure you guys understand, right? Go, 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 lots of you, lots of you. But back to the growth rapidly, that's where these come from. That's why you sometimes wake up in the morning and go, what the heck? Now that you know how a fungus really grows, the prominent part of a fungus, the part that the fungus is mostly made up of, now you know how it works. You could explain to this kid how that fairy ring, they call them fairy rings because they thought back years ago that you know, mythical fairies would come down and sprinkle, I don't know, whatever, something or other fairy mushroom stuff in a circle as they were dancing at night. And they would make these circles. But what would you tell this kid how come it's in a circle like that? And sometimes you don't get the full circle. I get them in my backyard. You get part of a circle, but you can see the arc. That'd be a good question. I may not ask it though because we're talking about it. What would you tell that kid? Why does that work? If you can draw on the screen again, you could. If you need to draw, you could show me. No one? Yeah, there you go, Spencer. Yeah, right there, right? Let me get white, be easier. So a spore germinates right there, and the mycelium mat grows out and grows out, like a web or netting, right? And you know what's going to happen is when that time for that might, you know, that basiocarp to form, all of those dikaryotic mycelia are going to start sprouting on basiocarps, toadstools, all in a circle. And of course, all you're looking at in that fairy ring, which is kind of cool if you were a mycologist, somebody would study that thing. You could use that if you knew where that first spore that Spencer pointed out, if you knew where that started, okay, and you knew when that started, you could get an idea of mycelial growth, the rate of mycelial growth. That'd be easier to do in a laboratory, of course, than in the wild. But I'm sure that's exactly what they've done to be able to calculate that. Fungus can grow pretty fast. Now, this part grows up really fast, right? Pop up. Time to go, guys. Fairy ring's a better word than mycelial extension. Yeah, absolutely. That's why you wake up and go, whoa, where did all those come from? Especially after a rainstorm. It's like I love going out in the woods. They're just like, these things go crazy. You're like, whoa, they're everywhere popping up. That's why, you know, with the hyphae that they have, even if you're septate, you still got pores. You can run that cytoplasm through real quick. So you're basically, what you're doing at the edge of that mycelial mat is you're plumping it up. You're forcing fluid and it's going, and it pops up above the ground. Like if you, like a sprinkler. Oh, great analogy. Like one of your sprinklers that pops up. It's the water pressure that pushes it up, right? Okay, fungi in the environment. Like I said, they break things down. That's good because they're good recyclers. They can act as mutualists, right? They can work in symbiotic relationships, right? They can, you know, help out with plants, obviously with the mycorrhizae. They can form, lichens and algae and cyanobacteria and fungi together. There's good fungus for animals too, right? There's examples where fungi are beneficial for animals. Like, for example, we'll talk about some ants here in just a second where the fungus is beneficial. You eat them. So, I mean, some of you might. My dad, I don't eat toadstools. I'm like, okay, Dad. They can also, the ones we hear about too, the parasites, the pathogens, they also exist. So we've talked about this before, but I'm not sure, I might have, because sometimes I do repeat things I talk about in lecture. But have I given you this chart before? This little one down below of how to, yeah, I have, good. So just remind you of that, okay? That's a good way to kind of remember all the different symbiotic relationships. So, okay, let's look at the plus ones, the mutualism ones. First of all, the mycorrhizae. I don't need to spend any more. There's some cool, look at those. Endophytic hyphae. All right, so that's endophytic hyphae is the hyphae growing out into the environment. I guess that's got to be a water bubble right there, air bubble, not water bubble. Endophytes, which would be bacteria, or I'm sorry, which would be fungi that live inside of plants. Like I said, oftentimes these are the arpuscular mycorrhizae, the ones that the ascomycetes and the glomeromycetes make. Found in all plants. Endophytes are found, whether they're mycorrhizae or not. There's fungi, mutualistic fungi that live in all plants. It's not surprising. They can get in all kinds of different areas, right? Herbivores have this advantage over their prey. Their prey cannot run away. If you're a grazer, that grass blade that wants to protect itself from you eating it, because of course when you eat it you kill it, it's a living thing, can't run away from you. So one of the things it can do is make it unpalatable for you to eat. So for example, if the fungus lived in a mutualistic relationship with a plant and the benefit it gave to the plant was that grazers and didn't eat it anymore, well, that would be obviously selected for, wouldn't it, right? In natural selection, wouldn't that be selected for? If you protected against a disease, if you were some fungus, any of those sorts of things. That's why we spend so much time on evolution, right? I said this whole class is nothing about evolution. If you produce some toxin that made herbivores sick, they'd stop eating it. As I said, the digestive, what we call the flora of your gut, that's the bacteria and the fungi that help you digest food. These leafcutter ants, what they do is they do something called fungus farming. They're actually, like we do, we're growing fungi. What they do is they take these leaves, they cut them up, they use their mandibles. They chop up these leaves and they carry them, right? Remember, an ant can carry, like, I don't know, 20 times its weight or some incredible disproportionate amount, you would think. The They take it to the fungus. The fungus digests the leaves, okay, and in return, now you think, how would the fungus do this? Again, it's all just, differential reproductivity. It's not a thought process. The fungus secretes this very nutrient-rich liquid at the very tips of the hyphae, which the ants feed upon. Okay, so I'll give you leaves to feed upon fungus, and fungus will give me back these little droplets of these very rich nutrient tips at the of the hyphae. Nibble, nibble, nibble. Okay, speaking of mutualistic fungi, sometimes they're obligate, right? Sometimes they have to happen. So we're back on the lichens again. Notice I put species in quotes, right? Just to highlight, or just to confuse you guys even more. Remember, they're not that. Well, they are species. They have names, but it really sort of pushes that definition, because remember, it's a mutualistic relationship. It's usually a cup fungus and then something that photosynthesizes. And then they help each other out, right? The thing that photosynthesizes makes food, okay? And some of the food it makes for itself, and the rest the fungus is able to absorb, right? It's the same thing like with the mycorrhizae. And then the fungus, back to the photosynthesizers, provides carbon. Remember, we're all a carbon-based life form. They reproduce, lichens reproduce asexually, forming these things called ceridia. The matchstick, or the British soldier lichen. Oh, I got to show you this, because I saw some out in the woods the other day. They didn't have these little things on them though. But hold on, match. I'll give you the link to it. Matchbox, Steve. Matchstick. Holy cow. These are really cool. They grow in Florida too. And we saw the fungus. Take a look at this thing. What we saw the other day, those are ceridia, the red tips that you see there. That's why it's called matchstick or British soldier. But the way we saw it the other day out at South Pine Lakes Preserve was like this. Okay. Oh, at the end of those little branches that you can see in that second picture, that's where those red tips would form, and those are the ceridia, and they would then branch off. Okay. You guys see those pictures? Can you that link it, or is that link working? Yeah. Okay. What do we mean by a pioneer species? Okay, a pioneer species, in spite of the fact that the lichen looks like it's relatively kind of a sensitive organism, it's one that actually can do well in what we call pioneer environments. Pioneer environments would be like when the plants moved out on the land. The critters that can do well in environments that are kind of sparse, that have ways of maybe extracting everything out of the environment in which they're found, no matter how harsh it might be. Like something like this would be able to do, right? Because of the fungus, huh? The fungus would digest any of that stuff, and that would benefit, obviously, the photosynthetic part. And since there wouldn't be, a lot of food or enough nutrients or energy molecules to absorb from the environment, the photosynthesis part would really help too, wouldn't it? So these were important even way back 420 million years ago. Now, even though it seems like an oxymoron, but even though they're really good at living in harsh environments, they're also in many cases what we call an indicator species. Now, what an indicator species is, let me see, maybe we talk about that later. Hold on before I do. I did have another picture of it. Oh, no, I guess I don't. An indicator species, the canary in the coal mine. Cold. Why did I say coal? Coal. Canary in a coal mine. You guys know what that means? What that refers to? That's not spell canary. Yeah. Sting sings about it in one of the songs from the Police. Canary in a coal mine. Do you know why that? You guys heard that phrase before? Yeah, maybe. They don't use them anymore, of course, for obvious reasons, because the canaries get very upset about this. But as we're going to learn about birds, birds breed in a very, very, very efficient way. They have to, because flying. That's true. Well, it can be in the coal mine, but here's the problem. Because they have such an efficient way to breed, birds do. When we talk about birds, we'll go over it again, but birds basically, in a very simple form, they breathe their air more than once. So in other words, they extract everything, oxygen really. They extract as much oxygen out of the air as they possibly can because they have such a high demand because flying is a very heavy metabolic activity, okay? So their lungs are designed in a really, really cool way that we'll talk about. But since they extract everything out of the air, that means they're going to be very good at extracting oxygen and extracting, bad things too. And one of the things that coal miners have to be careful about that accumulates in coal mines is carbon monoxide, right? The stuff that comes out of the tailpipe of a car, and methane. Methane and both carbon monoxide can cause, problems in breathing, obviously. Methane's also explosive. So if you put a canary in a coal mine with a bunch of humans who are just, let's say, regular breathers, right? Who's going to feel the effects first, the bird or the coal miners? Sure, the bird dies. Everybody get out of the mine. Bird dies. Must be something wrong with the air. So that's what we mean by an indicator species. Lichens are that way. In other words, what we mean is if the environment is changing, oftentimes an ecologist, when he goes out, if he's trying to document that a certain environment's being affected by, let's say, pollution, maybe stuff up in the air, air pollution, obviously what I meant, the lichens will be the things that die off first. So if he goes out into a stand of trees, a group of trees, and he documents some lichens over the years, and he knows they're going away, that might be an indication that it's, there's something changing in the environment. Even before the trees start dying off for the animals, the lichens are many times one of the first ones to go, okay? That's what we mean by that. Canaries in the coal mine. All right, I need you to know 3 types of lichen for the lab practical. I'm not asking you to identify them by species. I do want you to know them by identify them by species in your field notebook as well as you can do. But there's 3 easy ways to remember them. The crustose type looks like crust. Okay, that's like the stuff that looks like paint on the trees. The greens and the blue greens and all those colors that you see all the time. Foliose, this is foliose, but maybe a better way, the foliose that was in class actually. Foliose, a folio is a sheet, okay? Like if you had a portfolio, you'd have many sheets, okay? So foliose, we say, is leaf-shaped, right? Shaped like a leaf, which is shaped like a sheet, right? So foliose lichen. And then the other type is the bushy type, the fruticose. There was another type on lab, but don't worry about that type. Just the crustose, the foliose, and the fruticose. Questions on those? Everybody knows not a moss, right? You're not going to give me deer moss as one of your moss in your field notebook. You won't get any points for that at all. There's the one of the red ones. That's a crustose. These are Florida types. Baton Rouge lichen. Baton Rouge means red stick in French. I do remember that. Xanthoparmelia. I was talking to someone the other day in class when you're identifying things like, for example, sphagnum, that moss that I've mentioned to you guys a couple times. If we don't know the species, and sometimes telling the species is really difficult to do. Now, if you just can't tell the species, you can't use this. But if you look on the expert pages and you see this SP, that means that that's the genus, but they're sort of unsure about the species. And in some cases, when you're identifying your critters for the field notebooks, I'm okay with that. Now, if you have any questions about that, let me know. For example, in sphagnum. Sphagnum SP is fine. Xanthoparmelia is also another one. Deer moss is not. I think we have more than one species of deer moss too. Okay, let's look at the bad side. Fungus can also be parasites, and they can be pathogens. For example, they can affect chestnuts. We used to have a lot of chestnut trees in Florida, but then something called the chestnut blight affected trees. About 30% of fungal species are parasites or pathogens, so it's a lot like the prokaryotes, right? You're not going to really hear about them unless they're causing trouble. They attack food crops, removing 10 to 50%. You can imagine a farmer is going to be upset by that. The chestnut blight, look at the way it's spread out. Chestnut blight was introduced initially from, I don't remember where it came. It was a ship coming into the port, but in New York City in 1910. And begins killing chestnut trees. Those are what chestnut trees look like. You may have never seen one. There's a few scattered groups of chestnut trees, but notice by 1940, how far they were gone. 50 to 99% were dead. So in what, 30 years? See how quick that spread out? Wiping out the chestnut trees had no natural defense against this against this parasite because it came from a part of the world that the chestnut trees weren't found, you see. The poor folks, I think I mentioned to you guys in class the other day, the poor folks that they roasted at on the fire during the Salem witch trials, there are some thoughts, and in some cases, the erratic behavior of those folks that caused them to be thought of as witches might have been caused by hallucinogenic trips from eating ergot, which is a fungus, a fungal parasite found on rye, which they made bread out of. And the hallucinogenic they found that forms, or ergot forms, this hallucinogenic material very similar to LSD. So those poor folks may have actually just been experiencing the effects of LSD from the unknown, obviously, from the ergot that was growing on the rye bread. That's how horrible that must have been. I never knew how to say this stuff, that smut that's grown on the corn. That's the delicacy. It's a delicacy among many Hispanic dishes, and it's called, I got the abbreviation, huitlacoche. I've never had any of that, so if you have, it's supposed to be really good. It looks awful. But that the name above is the scientific name for it, but that's what we mean by a smut. Now that's a delicacy. So that's a fungal parasite, but it's not a pathogen to us. Now, some are toxic. I used to always tell my wife, oh, you can tear the blue bread mold off bread and still eat the good part, until I was reading that they found that the bread mold even secretes toxins into the bread that doesn't, even where the mycelia aren't found. So it's like, oh, maybe that's not a good idea. Frogs. Oh gosh, about a third of the world's population has been affected by this particular parasite. The name of it up above, Batrachotrichium dendrobatidis. Dendrobatid frogs are the poison dart frogs, real brilliantly colored ones. But it forms this pocket. You can see it in the stomach of this dead frog over here on the right. It's a chytrid. Okay, so it's Chytridiomycota. It forms this pockets, and the frogs die, or the amphibians die, for something called chytridiomycosis. The red spots, and sort of like we saw the spread of the chestnut blight, show you how quickly this thing is spread. Look at that. Reported around Mexico City, 1974. By 2004, it's all down by the Panama Canal, and then it's been spread all over the world. These are confirmed in red, not confirmed, but believed in blue, and I think unconfirmed in just reported in white. 200, almost 200 amphibian species are declining or on verge of extinction, or extinct because of chytridiomycosis. Now, if you think, well, what good are all the amphibians? Remember, it's an ecosystem, right? Everything all fits together. If you get rid of all the amphibians, one of the things we're going to have to deal with is a lot more mosquitoes. Because amphibians, when they're tadpoles, right? When they're in that tadpole stage, one of the things they eat a lot of, considering where they're found, is a lot of mosquito larvae. Everything all balances. Everything has a purpose. Everything balances out with everything else. Now, humans, we also have problems with this stuff. We suffer from things called mycoses, or a mycosis, which is some sort of a fungal infection. Candida, which is the critter that you see under the microscope here. Candida can cause what's called thrush. which is many oftentimes found in vaginal infections. Candida is actually a type of yeast. So it's very good at asexually reproducing, reproducing very quickly. Remember, asexual is very fast. You don't get variability, but if it's a good environment, which, without moist, dark environments are good environments for it, you're going to get a very rapid growth. And many times these are opportunistic. And what we mean by that is if you have a healthy flora in your body, whether in your stomach, like we talked about, or say in your vagina, if you have one of those, then you're protected from stuff coming in from the outside. One of the things that protects, one of the vulnerabilities in our body are any openings, okay? Cuts and wounds, or your eyes, your tear ducts. So many times you have enzymes and you have certain mutualistic fungus that live there, and certain mutualistic prokaryotes even that live there that actually are beneficial. They prevent other bad pathogenic fungi like Candida moving in. Now, if you're on antibiotics, right? If you're on antibiotics and you're female, you may have noticed that oftentimes or females will get a yeast infection soon after coming off antibiotics, right? Because what they've done is they've lowered the biotic, or specifically antibiotics are going after the prokaryotes, lower the prokaryote fauna or flora, excuse me, that was actually getting rid of some of these pathogenic fungi, and so you get a yeast infection. Folks who often die from this rare form of pneumonia, if they have AIDS, it's caused actually by this fungal organism, which the name is Pneumocystis jirovecii. All right, and it's a rare form of pneumonia. And I talked about this, I think, before about immunity, right? You think, oh gosh, this poor person had AIDS, and then they encounter this rare form of pneumonia because maybe it's just not very, frequent in the environment. Well, it actually doesn't mean necessarily it's not frequent. What we mean by a rare form of pneumonia may be a type of pneumonia that only affects people with compromised immune system, or affects people more with a compromised immune system. Okay, so if you didn't have an AIDS compromised immune system, you might not suffer from this rare form of pneumonia. So, okay, athlete's foot, right? If anybody in Florida, when your feet get wet, if you don't take care of them, you get athlete's foot. Always wear flip-flops in a common shower. Okay, that's one of the ways it spreads. Ringworm, which is not a worm, but makes these little red marks. It's actually, there's a microscope shot of ringworm, forms a little pocket in the skin. Nail fungus, which you don't have to worry about till you get older. Thank goodness I don't have. I think, it just grosses me out. I have this weird thing about toenails. Cannot stand gross toenails. I don't let anybody cut or even touch my toenails. I don't know what it is. Yep, if you lay down, a friend of mine got it from changing the tire on his car when it was raining. He had to lay down in the wet grass for something, and he got it there. It's not hard to control. You go to the doctor, and there's a topical fungicide they give you. We all like cheese, right? We all maybe enjoy beer now and then, and we all like bread. It's interesting how alcohol and bread are both made kind of the same way. Anheuser-Busch actually, during the Prohibition, switched a lot of the production over to the making of bread, since it's a similar process using yeast and fermentation. Yeast releases the carbon dioxide, causes the bread to rise. Okay, so we saw yeast the other day. Remember, these are the Saccharomyces cerevisiae that we looked at. And I said, isn't that name cool? Right there, they're budding right there, aren't they? Penicillium, which was like a string of spores, asexual spores, Conidia spores, on Conidia fours. Now, useful for us, of course, for the stuff that we eat or drink, but also from a medical point of view, right? Penicillium, the fungus Penicillium, I think we had some on our prokaryote plates that were growing. Actually, if you try to grow Staphylococcus, if you try to grow one of those grape-shaped bacteria, you get this, look at that. They don't grow right there. The zone of inhibited growth. That's how it was discovered. The dude who discovered penicillin's antibiotic properties, he had a petri dish that he put on a windowsill in the lab where he worked, and the window was open. And I can't remember if he left the top off or whatever the case may be. Throughout he was growing some bacteria on it. about the weekend or however long he was gone, this fungus from the outside, penicillium is naturally occurs in the soil. It's a natural occurring fungus. Landed on the petri dish and grew. And when he came back, he said, wow, notice how the bacteria don't grow anywhere next to it. And voila, you got penicillin, right? The fungi are also important in genetic research, doing genetic engineering, right? Remember I was telling you how in prokaryotes you can inject the gene for insulin, even though they don't use insulin. You can do the same thing with fungi, especially like yeast, right? You could inject that gene into yeast, get them asexually reproduce, bam, 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 make a lot of yeast. They unwittingly, like bacteria did, make insulin. Got yourself a bunch of insulin, right? That's what we mean by genetic engineering. Genetic recombination, same thing. Okay. I know we only got 10 minutes, but I can't wait. So it's kind of like a kid before Christmas. I've been thinking about getting into this chapter all semester long, so I do want to get started on it. This is absolutely my favorite. What does the last line in the PowerPoint? Whoops. Let me go back. Hold on one second, Thomas. Sorry. Got so excited about it. Oh, insulin-like growth factor. Okay, so what we're saying is if you can genetically engineer it to produce insulin, if you give it an insulin-like growth factor, then it's going to produce tons of insulin. Remember, growth factors are molecules that stimulate something to happen. If you take an AMP or go on to take AMP, you'll talk about lots of growth factors. Talk lots of factors. Now the animals, our last chapter. So it's 3 chapters long. The first chapter is relatively short, where I kind of just talk about what makes an animal an animal. And then in the other 2 chapters, we're going to actually look at the different kinds of animals. So I picked this really cool background, right? Scales of a snake. If I hadn't studied fish, it might have been snakes. I'm fascinated by snakes. Does anybody remember what this is? I know it's a song. You might remember the song. Look how clean cut they were dressed. They're probably all doing LSD and smoking marijuana like everybody back then. But look at the Beatles. Remember the Beatles when they first came out were all clean cut? So do you know what would be inside this square thing? I'm being facetious. Finally got rid of all of mine the other day. Well, not the other day. The other year, my wife said, I'm not looking around your records anymore. Nobody has that anymore. Nobody plays records. Yeah, exactly. Even CDs. What? CDs? Holy cow. Oh goodness, oh goodness, oh goodness. Where do I start? Yeah. Some people say, some purists say that the sound is more pure on a record. Okay. You know, I don't know. I guess if I could hear like a dog, maybe, or maybe if you're a music aficionado, right? Gosh, where do we start about animals? Where do we start? First of all, let's talk about how they make their living. The bear got to eat, right? So that's what we talked about a long time ago in prokaryotes. It's chemoheterotrophy. Get your energy and you get your carbon from another source, not like the plants. Finally, multicellularity in the sense of what I've always been kind of referring to as tissues. Okay, so plants, if you look somewhere, you'll see, for example, the xylem, the phloem, the cambium, the bark. You'll see those referred to as plant tissues. And I'm not trying to say that they're not, because even among animals, because when we get into the porifera, the sponges, we find that they're really not tissues either. Even though they're considered animals, they're really more kind of a loose aggregate. But it's the animals that raise this idea of multicellularity to the next level. Don't get me wrong, cambium and bark and xylem and phloem are all important tissues in plants, but man, you're talking about in animals, they start forming tissues like muscle tissue, like nervous tissue. You don't find those kinds of tissues in plants, right? Plants don't have muscles. Plants can't think. So animals have really polished off this idea of cells working together. Cells working together in a tissue, not just as an aggregate, not just like the Volvox, not just, you know, what was one of your questions in lab, is Volvox a collection of cells or multicellular? And it's kind of like, yeah. These guys take the idea of cellularity, these animals, us, take this idea of cellularity to the next level. They make this structural unit, this functional unit. Look at this really cool picture. This is a muscle cell here, right? This is a nerve cell. coming down and right at the spot. We call it, we call it a neurosynaptic cleft that occurs between nerve cells. Oh, I'm sorry, nope, a neuromuscular junction. Excuse me, got to remember my A and P. Where I pointed with the arrow is called a neuromuscular junction. It's cool, folks. This is right where the electrical impulse from the nerve, the wire, right, stimulates the muscle cell, makes the muscle cell twitch, contract. It's all chemical. All chemical. It's all this great, incredible, beautiful chemical dance that occurs in living things, right? We don't have cell walls. Don't have cell walls. Now that means you got to have a skeleton on the outside or on the inside, but right, that's a cost. So you got to get one of those. Worms don't have an exo or an endoskeleton though. They have a hydrostatic skeleton. What's a hydrostatic skeleton? Put your thumb over a garden hose. The garden hose tightens up behind it, right? That's what we mean by hydrostatic skeleton. That's how worms do it. But not having cell walls is nice because we're flexible. We can move around. And one of the things being flexible allows you to do is you ain't limited to how much water you can cram into your water vacuole anymore, right? You can go get water. Now, you can move fast. You can move slow. You can fly. You can swim. You can walk. Not everybody. Not everybody. Some folks are sedentary. Okay, some folks are sessile. Some folks don't move around. Some animals move around during certain parts of their life, like when they're a larvae, might drift off, right, for dispersal reasons, and then they settle down. That's how corals, for example, do when we start talking about them. Corals make a larval form that drifts off and then settles down into a coral for the rest of its life. Isn't that a cool picture right there? Those are animal cells just getting ready to finish cytokinesis. Remember from mitosis? See, the whole idea of tissues, the whole advancement of tissues is due to a protein that you learned about back in Bio 1. See if you guys remember this protein, collagen. Where would you find collagen in your body if I asked you to, point to a piece of, or an area of collagen? Where would you point to? Yeah, that's one of the places collagen forms. There you go. Skin. Yep. Lobes of your ears, lips have a lot of collagen. Some folks get, beef collagen injected in their lips. Oh, just thinking about it. But it's a stretchy, flexible, sticky kind of protein. Plants don't make collagen. Animals make collagen. Making that collagen allows cells to stick together. Don't need no cell wall. Don't need cellulose. I got collagen. So I got this sticky, stringy stuff that I can start clumping these tissues together to form these really, really, really cool structural units. The muscles, the endocrine system, right? The chemical message system. It just fascinates me. Anyway, all kinds of different animals, right? All kinds of different. It's like when I got into biology, I'm like, oh my goodness. But sharks always interested me first. So it was always oceans and fish, right? A difference in how they look. All the way from this millipede thing, if you kind of wondered how you know between a millipede and a centipede, we'll talk about how that works. Some that only exist in the ocean. So there's over 8 million species, 35 to 40 phyla of animals, pretty diverse. Many of the phyla only found in the ocean. For example, these guys: brittle stars, sea stars, sand dollars. Sea biscuits, sea urchins, right? You guys are familiar with all those, right? Only found in the ocean. No terrestrial forms. No terrestrial forms. Cool ways of getting food, like the chameleon with its tongue, reaching out and grabbing that. Oh, God, that's so cool. And their eyes. Oh, love those kind of critters. Sexual reproduction, which sometimes involves internal or external fertilization, no matter how fast or slow. Gosh, I remember years ago being at the San Diego Zoo, and there were Galapagos tortoises that were out there mating, and there was this huge crowd of people gathered around, and they were hooting and hollering. I was thinking, oh my goodness, if only reptiles could think. What are you people looking at? Get a room. That's everybody, just awful, folks. No alternation of generation. No alternation of generation. These critters. In animals, we don't do it the same way they do. We're gonna stick with the unicellular gamete. It's time. Let me got one more thing to say. The way we develop, I got two more things today. The way we develop embryonically is very unique, and that's what you're seeing down below is a little ball of cell. Talk more about that later. Albatrosses, males, 6-foot wingspan, that bird on the right-hand side. Male albatrosses rarely, if ever, touch ground after they're born. Just wanted to finish off lecture with that. When they mate in the air, male albatrosses mate in the air. Females land when they lay eggs, but males can, now they land on the water, and they fish out of the water, but they rarely ever touch land in many cases after they're born. Okay? All I want to say, that's your thought for the weekend. We'll continue on with animals next week. So if you don't have any questions for me, I'll talk to you all later. If you have questions, then I'll stick around and answer them for you.